What's going on, guys? Welcome back to another breakdown. We're going to be doing a BB passage. We're going to be doing BB passage number... Which one didn't we do? I think we didn't do 7. This is 5, 6, 7. Okay, we're going to be doing passage number 7, guys. Not going to lie, I'm a little under the weather today. So, I'm going to push through, though. Okay, so I'm going to show you guys how to highlight, how to pick the best answer, how to make all these details make sense, how to interpret figures, how to interpret graphs, everything like that, so you guys can get that 132. All right, I'm gonna show you guys how to do this, especially in the best amount of time possible. Okay, I'm gonna make sure you guys do this in the least amount of time. All right, the pace that I read these passages should be the pace that you should be going. Okay, the pace is a regular reading pace, but a little slower. Okay, just like you would read a book or something, but just a little slower so that you could zone in on these details. All right, so as always, guys, do this on your own first and then hear me break it down. So this is passage 7. This is pause it whenever you need to. This is the passage here. All right, read through it, answer this question, write down your answer. Write down your answer, pause it when you need to, write down your answer, and write down your answer. This one, that's it. Okay, we got 46 to 51. A lot of questions here. All right, let me begin now. Follicle stimulating hormone is a peptide hormone known to activate FSH protein coupled receptors on ovarian cells. Nothing to highlight here. I know all this from my content review, so I'm not going to highlight anything. However, FSHRs have recently been found on osteoclasts, and their activation stimulates osteoclastogenesis and bone resorption. Okay, so FSHRs, they're found on osteoclasts, all right? And when FSH binds to FSHR on the osteoclast, you get bone resorption. They get activated. Although estrogen activity inhibits these effects, high levels of FSH despite normal levels of estrogen are associated with bone density loss. All right, so I'm going to highlight estrogen activity inhibits these effects. All right, you want to highlight whatever sticks out at you. You highlight it because the question will most likely ask about it. All right, do more passages and more practice, you'll get better at identifying what sticks out. Okay, so if I were to write something on my notebook or that crappy dry erase paper they give you on the MCAT, all right, and the crappy Sharpie they give you, this is what I would write. Okay, it's a quick note. And usually in the BB passages, you'll see me write more on the dry erase paper than the CP passages. CP passages, the only time I'm writing is to do math. That's it. So for BB, you want to write something quick and you want to use arrows and equal signs. Okay, so how would I do this for this? I would write high FSH equals overcomes the estrogen block. All right, that's what I would write. That's it. It's that simple. You may write something similar, a little different, but as long as you get the main point here in that passage, in that paragraph, was that estrogen blocks the osteoclastogenesis, but high follicle stimulating hormone can overcome this estrogen block. Right? A peptide consisting of a 13 residue sequence located within the receptor binding domain of FSH was synthesized, FSH PEP. Okay, so FSH PEP was the one that they synthesized. An antibody, FSH-AB, was then designed to bind the specific sequence of FSH PEP. The antibody was also able to bind FSH. So I'm highlighting this. This antibody can bind FSH and bind FSH PEP. And this is the structure. Don't look at these figures. Don't look at the figures yet. Only look at the figures when the question asks for it. If you look at the figures and try to interpret it before the question asks, you will waste time. Okay. The specificity of FSH-AB for either FSH PEP or FSH was compared using an enzyme-linked immunosorbent assay. ELISA. Okay. You guys should be familiar with ELISA. You should know the process of ELISA and what it does. All right. In which an enzyme-linked antibody was used to detect FSH-AB that had bound either FSH PEP or FSH. When enzyme substrate was added, a fluorescent product formed. The intensity of the fluorescence was measured, and increased absorbance was indicative of increased binding. Okay, so they were nice to you here, and that they told you that increased absorbance meant increased binding. Okay, you might have not knew that if you didn't know what ELISA was. All right, but they told you here. So I would write in the whiteboard real quick. Boom, increase fluoresce equals increase bind. That's what I would write. It doesn't even have to be legible. Okay, these notes are quick. You don't want to waste time. This is a figure. We don't look at it yet. Let's go to the question. Based on the passage, FSHRs are found on cells of which type of tissue? Well, they told us it was on bones. So bones are a type of connective tissue, and they told us it's in the ovary, all right, ovarian cells. And so in the ovarian cells, you're going to have some epithelial. You're going to have epithelial tissue, all right. I think there's some, I don't know if I would consider the ovaries connective tissue. All right. I know for a fact that these two will be there. I don't think nervous tissue would have FSHRs. It would not. Nervous tissue would not. Okay. So connective and epithelial is the answer. All right, 1 and 2 here. All right, connective tissue is like your bones, your muscles. All right, connective. It's connecting your body. Epithelial, think of like the lining of internal organs and stuff like that. All right, and your skin, internal and external organs, the lining. That's epithelial usually. Oh, I didn't put the answer thing. Okay, so the answer for this one, C. And that's it. We got it correct. Every time I answer a question, I'm very confident in my answer. You guys have to be confident. If you're confident, it's going to prevent overthinking. Overthinking is like the big enemy of a lot of students, all right? Which conclusion is supported by the data presented in figure 2? Okay, when a question asks you something, you hang on to every word. Which conclusion is supported by the data presented in figure 2? Sounds common sense, but you go to figure 2 and you only go to figure 2, okay? So anything that involves something outside of figure 2 is wrong, okay? Anything that involves a reach outside of figure 2 is wrong as well. All right. So A, the KD for binding of FSH PEP by FSH-AB is lower than the KD of binding of FSH by FSH-AB. So what has better absorbance? Whatever has better absorbance was better binding. All right. What's better binding? FSH-E PEP or FSH regular? If I look at here, I see that FSH PEP's absorbance was like a high. It's pretty high here. And if I look here, FSH absorbance is high, but there's a different scale. All right. This is like a 2.7, 2.8. This doesn't even reach 2.8. This is 0.1, 0.2, 0.3. So FSH PEP has better binding, has more binding than FSH. All right. And because FSH has more binding, the KD is lower. Remember. All right. A high KD means low binding. And then a low KD means high affinity. Okay. Low KD, high affinity. High KD, low affinity. So therefore A is correct. And A is proven. It's 100% supported, 100% proven. It's correct. All right. But I'll go through the other ones to see. IgG has a higher affinity. No, we're not looking at IgG here. It doesn't even have a higher affinity. Look how small it is. That's IgG, right? Yeah. FSH binding sites for FSH-AB are saturated when FSH reaches a concentration of 10 micrograms. So FSH-AB, AB are saturated when it reaches 10 micrograms. I don't know that. Okay, it does increase, but maybe it increases more. It keeps going at like 12, 15. I don't know that. So this is wrong. This is wrong, I said. The affinity of FSH for FSH-AB is greater than the affinity? No, it's lower. Okay, it's lower. Therefore, the answer is A. I'm very confident it's proven. I already know that's correct. Let's keep going. That's how you do it, guys. But bam. Which statement best explains why the absorbance levels for FSH differ from those of FSH PEP? The FSH AB binds non-specifically to more than one site on FSH PEP. This is wrong, okay? The FSH AB binds specifically to one site only. How do I know this? Well, the whole point of this whole thing was constructed to bind FSH PEP, okay? The FSH PEP was created and the FSH antibody was designed to bind FSH PEP. All right, so this is wrong. IgG competitively inhibits the binding of FSH-AB to FSH. So let's check that. Here's FSH. Okay, this is wrong. B is wrong, because if this were true, you would see more IgG would equal to less FSH-AB. But instead you see relative the same amount of IgG and an increase of FSH-AB. All right, you would see something different. You would see IgG and then like a very low FSH-AB. All right, so this is wrong. FSH-AB cooperatively binds FSH. This is also wrong. If it was cooperative, I would see some type of growth that was somewhat exponential, a huge growth. Okay, this looks, and I would see a sigmoidal type of growth here as well, you know, but this is more linear. Look, it's going up, more micrograms, more FSH-AB. All right, it's going up. Let's keep going up linearly. All right, so I wouldn't say this is C. The tertiary structure of FSH limits FSH-AB binding interactions. Process elimination, we're going to go with D. Do they say that this is just a primary structure here? All right. Pass 13 residue sequence located with the receptor binding domain. Maybe this is just for this 13 residues. It's looking like a primary structure, and then FSH is actually the whole entire protein coiled up, condensed, everything. All right, so yeah, D is the answer here. All right, I'm not crazy. I'm not doing backflips and saying that this is right. However, process elimination, I'm picking the best answer, not necessarily the amazing, backflip. I'm screaming that this is correct answer, okay? So, process elimination, this is the best answer, so we're going to go with D. Okay, you don't have to feel amazing about every single answer choice. MCAT's not going to expect you to feel amazing about every answer choice. Okay, MCAT knows what it's doing. MCAT words, it makes these questions with great detail. They put a lot of thought into these questions, right, and answers. They know what they're doing. Okay, they know what they're doing, and we don't even know what we got right and what we got wrong in the MCAT. Okay, so they're playing something out there. Based on the FSH PEP sequence, which amino acid substitution in the FSHR binding domain is most likely to have the greatest effect in reducing bone density loss in the presence of high levels of FSH? Okay, well, they want to disrupt. Okay, let's look at this here. Okay, I can't talk today. I'm sorry. They want to disrupt this. They want to disrupt the binding. If you disrupt the binding, you're going to have a reduction in bone density loss. Okay, why? Because if you disrupt the binding, you're going to stop the activation of osteoclasts. Thus, you're going to stop the activation of bone density loss. All right, so what disrupts this? K replaced with an R, a lysine replaced with an arginine. Two positives, that wouldn't really disrupt anything. You have an arginine replaced with the aspartate. You went from a positive to a negative. That would disrupt. Valine to, no, these are both hydrophobic. They're pretty similar as well. Nothing charged here. The biggest drastic change here is B. So we're going to go with B. Okay. That's how you do it. But bam. Which of the following reasons does not describe why FSH PEP was included in the ELISA experiment? All right, let's see this. Hold on, guys. I gotta sneeze, but I'm not sneezing. All right. So let's look at A first. Okay, let's look at A. To act as a positive control to confirm that this say was functional. Okay, maybe. But I could see why. Okay, so if we look at this experiment, what's the point of the control IgG? No, the point of this is to be a negative control. Okay, if we see a lot of absorbance of IgG, what would that mean? That would mean that our antibody binds to IgG. Okay, we want our antibody to bind to FSH. So IgGs are negative control here. All right, making sure that this protein does not go and bind here. And FSH could be our positive control. Okay, because if the antibody binds to FSH and it binds to FSH PEP, let's say, oh, they're asking for FSH PEP. So let's say this antibody does bind to FSH PEP like it did. That tells me that this sequence they made was good. It was correct. Okay, it acted as a positive control in a way. If, let's say, that this antibody did not bind to FSH, let's say it did not bind to FSH PEP, all right, what would this tell me? This would tell me that our sequence here, we made something wrong. This is wrong here. Okay, the whole point of making the FSHAB was to bind to FSH PEP. If this is wrong, if one of these nucleotides are wrong, or we did something wrong in the experiment, that would tell us. So I like A in the fact that it would describe, why it was included. Okay, so this makes sense. So I'm going to eliminate this. To provide a baseline against which to evaluate the affinity of FSHAB for FSH. Yeah, that's what they can compare values using that. So yes, that is a great way. To generate data to support that the FSHAB binds to the receptor binding domain of FSH. This is very similar to A. All right. What A is describing C, and C is describing A. Okay. When you have two that are very similar, that almost mean the same thing, they're both wrong no matter what. To determine the amount of FSHAB that is most effective as a therapeutic treatment, I like this answer as the one that does not describe why FSH PEP was used, because in order for something to be a therapeutic treatment, there's a lot going on in that. You got to make sure that this is where to inject this drug. Do we inject it? Is it an oral form? How are we going to put it in the body? You got to see the half-life. You got to see the effect. You got to see a lot of different things. And we can't say that it's a therapeutic treatment just by this. All right. So the answer for this one is D. All right. Which statement describes a characteristic of FSH? This is straight content review. You should know this. All right. FSH does not require transport proteins to remain soluble in the blood. Yeah, it does not require them. It's a protein. It's soluble in the bloodstream. Okay. FSH enters the bloodstream by diffusing. No, only steroid hormones do that. FSH is synthesized from cholesterol. No, steroid hormones are. FSH is derived from a single amino acid. Maybe. I don't know. Okay, but I'm left with two answers, A and D. Okay, and think about what the better answer is. To find the better answer, there's three things I want you guys to keep in mind. Number one is whichever is more specific is usually the better answer. Number two, whatever is more proven in the passage is usually the better answer. And number three, whatever the MCAT is testing more is the better answer. The MCAT is testing our knowledge of biology, endocrine system, everything like that. Okay, the MCAT is not necessarily testing our knowledge of the derivations of hormones. They test us on how these hormones work and how they're transported, all that stuff. Okay, so because of that, A is the better answer, and let's see if it's correct. But bam, that's how you do it, guys. And that is it. All right. If you're interested in working one-on-one with me inside MCAT University, guys, is a fully loaded program. Okay. Plenty of students are going to hit their target score. It's guaranteed. It's absolutely guaranteed. It's very easy to follow as well. All you have to do is just follow the program, follow the proven path, and you will hit your target score. Okay. There's no difference between me and you. What worked for me is going to work for everybody. Okay. Also, I will make sure that you hit your target score in less than half the time. Okay, half the time. When you're doing the right things, you're going to see improvements very, very quick. Okay, very quick. So if you guys are interested in that, go ahead, click on the link in the comment section, fill out the application, book an interview, and if you're a good fit, then I'll ask you to join MCAT University, and you will hit your target score. So I'll see you guys in the next video. Subscribe. Let me know what you guys think in the comments.